You should have called me.
I've been forget on my cereal.
I might take some.
They had no fat at all. None. But their intestine also just tore apart by touching it. So.
See, that's just an idea.
I'm just going to give a little bit of an introduction and hopefully try not to embarrass Jess too much. I think I did that enough with my previous graduate student, Christian, who doesn't seem to be here. So maybe I'll show some more slides of Christian. So Jess joined my lab a very short time ago in my mind, but in the spring of 2009, after doing three rotations, and after kind of a friendly but heated struggle between John Blennis and myself, who both very much wanted Jess to join the lab. Even during her rotation, it was clear that she was a very talented young scientist. And despite the fact that I gave her probably 12 to 13 different rotation projects, and the fact that my lab had shrunk from 8 to 4 at this time, Jess made 5. This was about 4 years into my faculty position here, and the first round of people were leaving. My first round of funding had run out, and kind of we had about 10 grants pending, and Jess decided to join the lab despite all that. So hopefully things I think worked out okay. So this is at the bottom of a hike that we went on. Jess is an avid hiker. That's sarcasm. You have to know Jess to know that. So I have to show just a couple of pictures from this slide, some action photos. Jess did a fantastic job on the way up. Quite timid on the way down, which is really kind of quite the opposite of how she does science. She's very much not timid at all. Takes things head on, and really is a fearless and incredibly talented researcher. And I was very lucky that she joined my lab. So 3 years later, the lab has grown and completely turned over, and Jess is now departing as some new people are joining the lab. But between in that 3-year period, many things happened in Jess's life. The first of which is, and probably the most important of which, is she got married, not to me, but to this character here, Derek, who I think that a lot of people know. He actually was briefly and part-time was a technician in my lab for a talented graduate student in the lab. So this is some pictures of Derek and Jess, and really enjoyed getting to know Derek. The other thing that Jess was an important thing that Jess did over these years was she was really a stabilizing force between the different personalities in the lab. And this is my most senior graduate student, who's been out of the lab for about 3 months now, although he spends more time here now than he used to. And this is the newest graduate student, and you can tell Stefan could really go either way. We hope that he'll lean more this way, with having spent some time under the tutelage and under the guidance of Jess. So one of the most important things that happened over this year, and the most tangible thing that happened over these 3 years, was the changes to Jess's CV. I'm not going to go through every paper, but this is what her CV looked like in 2009. Her last name was not Yecies, it was Lucas, and she was by far not the most famous Lucas, as her mom, Judy, has had many, many more papers than her, and still does. But over that 3-year period, Jess made a dent and is coming closer to Judy's CV, and has really published a large number of papers over the past 3 years, has written really reviews that have already been highly cited, and has really made a great impact in our lab, but also in the field of Tor signaling in general. I joke because this top paper is also about mTOR, even though this is proof that she worked on TOR before joining my lab, but I always joke to Jess that she's been working on mTOR since she was either in diapers or at least in high school, I guess. So what Jess will talk about today are really her two main projects. One, which is kind of in vitro characterization of a new downstream target of mTOR called S3BP, and then an in vivo characterization of that same target. And Jess's projects really stem from some long-term projects that have been going on in the lab from my first two postdocs, Sui Zhang and Katrin Duvel. And Jess came into the lab and really, in a very fearless manner, took on these projects and made things that we were struggling with a bit much clearer and really made them her own. So she's co-first author in this first paper, and obviously she is first author on this last paper. And so I think that I'm going to let her speak. I think you're in for a treat, and I don't want to put too much pressure on her because she's an incredibly well-spoken scientist. Every time she gives a talk, especially outside of Boston, my inbox is just flooded with complimentary emails. So I think that she'll tell the story better than I ever would. And so we'll miss Jess. We wish her luck. Friday is actually her last day, so but really it's been a real pleasure having her in the lab. I'll hand it over to her.
Good afternoon, everyone. Thank you so much for coming to my dissertation seminar. As Brendan told you, I'm going to tell you about my work in the lab today about S3BP and its role downstream of mTOR1 signaling in cancer and in metabolism. So mTOR, mammalian target of rapamycin, is a serine 3 kinase. It is found in two distinct complexes within the cell, mTOR complex 1 and mTOR complex 2. Today I'm going to focus on mTOR complex 1, which senses growth factor and nutrient availability as well as cellular stress cues, and integrates these signals in order to control processes such as cell growth and proliferation. I'm going to focus on, in the first part of my talk, the in vitro studies that identified mTOR1's regulation of the transcription factor family, known as the S3BPs, and its regulation of cellular lipid metabolism as well as the pentose phosphate pathway. And then I will tell you in the second and third parts of the story about two in vivo settings that I followed up these observations in. First, in a metabolic tissue that does de novo lipogenesis normally, the liver. And second, in tumors, which tumor cells acquire the ability to carry out de novo lipid synthesis. And mTOR1 is frequently activated in these settings. So mTOR1 is apparently activated in a variety of diseases, including genetic tumor syndromes, cancer, and type 2 diabetes and obesity. There are a number of upstream branching signaling pathways that sense cellular cues such as stress and growth factor and nutrient availability, and many of these signals converge upon mTOR1's negative regulator, the TSC1-TSC2 complex. While we've come a long way in understanding upstream signaling pathways that regulate mTOR1 signaling, the consequences of aberrant mTOR1 activation are less well characterized. And given its importance in these diseases that I mentioned, our lab is very interested in understanding novel processes that this pathway controls. Here we'll focus in a little bit more closely on mTOR1's regulation by a small G protein switch that lies directly upstream. As I mentioned, the TSC1-TSC2 complex is mTOR1's negative regulator. It does this by exerting GTPase activating activity or GAP activity towards mTOR1's potent and essential activator, Rheb. When there are growth-promoting signals that want to activate mTOR1 in order to drive growth and proliferation, the TSC1-2 complex will be inhibited, leading Rheb to accumulate in the active GTP-bound form and stimulate mTOR1 signaling. When conditions are unfavorable for cell growth and proliferation, the TSC1-TSC2 complex will be activated, thereby converting Rheb from its active GTP-bound state to its inactive GDP-bound state, and thereby shutting off mTOR1 signaling. In the lab, we use a system in which you can genetically ablate either TSC1 or TSC2 to constitutively activate mTOR1 signaling and isolate it from many of those upstream pathways that I showed to you previously. This will lead Rheb to be GTP loaded constitutively and mTOR1 signaling will be on. This is evidenced on the signaling plot on the right. You can see that phosphorylation of S6 kinase, mTORC1's target, is shown as a readout for mTORC1 activation. We can compare wild-type, TSC1, and TSC2 deficient MEFs. What you can see is that in wild-type MEFs, if you deprive of serum, mTORC1 signaling is strongly suppressed. However, in contrast, in TSC2 and TSC1 deficient MEFs, even in the absence of growth factors, mTORC1 is constitutively activated. So this is a gain-of-function model for mTORC1 signaling that we can then combine with an inhibitor that is specific to mTORC1, rapamycin, under most conditions. And you can see that when you add rapamycin to TSC-deficient cells, you shut mTORC1 off again. So this is a system in which we can ask what the cell-intrinsic consequences of mTORC1 activation are, mTORC1 really alone and isolated from upstream pathways, as well as mTORC2 signaling. So in the next set of experiments that I'll show you, we use these conditions to interrogate the cell-intrinsic consequences of mTORC1 activation. Under serum-free growth conditions, wild-type cells will have mTORC1 in the off position, TSC1, TSC2 deficient cells have mTORC1 on, and then mTORC1 will be off again when you treat TSC1 and 2 deficient cells with rapamycin. So we perform parallel genomic and metabolomic approaches, performing a microarray to identify mTORC1 regulated transcripts and LCMS to identify intracellular mTORC1 regulated metabolites. What these two unbiased approaches demonstrated to us was that mTORC1 can control specific metabolic pathways. I'm going to summarize some of the results in the next section with a focus on SRBP. So this is a complex diagram here, but what this demonstrates is that when we looked at the microarray analysis of mTORC1 induced genes, those are genes that are elevated compared to wild-type cells and brought down towards wild-type cells when we treat with rapamycin. When we performed gene set enrichment analysis, we found that mTORC1 induced genes are enriched in 3 specific metabolic pathways. Those of glycolysis in blue, the pentose phosphate pathway in purple, and in orange, lipid and sterol biosynthesis. You can see highlighted in red are the mTORC1 induced genes that encode the enzymes in these pathways. You can see that mTORC1 controls expression of genes involved in glucose uptake and its glycolytic conversion to lactate, genes involved in both the oxidative and non-oxidative branches of the pentose phosphate pathway, as well as genes involved in the accumulation of acetyl-CoA and its conversion to sterols and isopromotes, as well as fatty acids and membranes, fluid membranes. So we were interested in how these genes are regulated downstream of mTORC1, given that mTORC1 is a kinase and not a transcription factor. Using a software known as Motifate, we did another bioinformatic approach to identify cis-regulatory elements that were enriched in the promoters of mTORC1-induced genes. The most significantly enriched transcription factor binding element was for SREBP, or sterol regulatory element binding proteins. So I'm summarizing data again, but Suman and then Katrin-Dubel in the lab demonstrated that glycolytic gene expression downstream of mTORC1 was due to increase in HIF1 alpha. What we found is that, as predicted by Motifate and cis regulatory element analysis, SREBPs are required downstream of mTORC1 for the induction of lipogenic targets. Importantly, we found also that the rate-limiting enzyme of the pentose phosphate pathway, G6PD, is also regulated by SREBP downstream of mTORC1. And this made sense to us because these two pathways are highly interrelated, where the oxidative branch of the pentose phosphate pathway produces NADPH, that is a reducing equivalent required for many anabolic processes, including de novo lipogenesis. So it's interesting to us that one transcription factor downstream of mTORC1, the SREBPs, could coordinate these two interrelated processes. So we wanted to know what were the consequences for cellular metabolism of the increase in expression of these lipogenic and pentose phosphate pathway genes. I measured de novo lipogenesis in these cells by labeling with C14 labeled glucose and then tracing the incorporation of C14 into the lipid fraction. What you can see here is that in TSC deficient cells, there's almost a 2-fold increase in de novo lipogenesis compared to wild type cells. And this is completely suppressed when you treat with rapamycin and inhibit mTORC1 signaling. When you knock down HIF1 alpha, there's really no effect on the increase in de novo lipogenesis. However, when you knock down SRBP1 in the TSC deficient cells, you strongly reduce the de novo lipogenesis rates, and this is completely and further reduced when you knock down SRBP1 and 2. The signaling plot below demonstrates efficient knockdowns, but also that knocking down SRBPs does not affect mTORC1 signaling, demonstrating that SRBPs are downstream of mTORC1 in the regulation of this process. So we're really interested in how mTORC1 is regulating SREBPs, and I want to provide a bit of background on how SREBPs are regulated, since it is a very complex process. This is a simplified schematic, but what you can see is that SREBP, in complex with the protein SCAP, or SREBP cleavage activating protein, resides in the ER in its inactive precursor form. SREBP SCAP is retained in the endoplasmic reticulum by a family of proteins known as insig proteins. And I'll talk about these in the second part of my talk. Cellular signals such as depletion of intracellular cholesterol